So I know I'm the only person left standing between you and that awesome photo and some lunch, so I'll make it pretty brief. I'm Laura Prentis. I'm a graduate student in the Robert Rizzo lab, and we are a computational drug design lab, not the Breaking Bad sort of drug design, more of like the cancer therapy drug design. And we also are one of the main developers of Doc6, which is one of the oldest docking softwares. And so in terms of drug design, viruses and bacteria are kind of like trying to treat like a moving target, and a pretty fast one at that. Evolutionarily speaking, they're pretty clever. So you can spend all of this time finding this amazing drug, and Ebola will come in and be like, nah, we're good. And it'll figure a way around it. So it can be very frustrating to try and treat these things. Think of HIV, for an example. It actually might be one of the most well-studied viruses. In fact, we know so much about HIV that it helped elucidate mechanisms for other viruses like Zika. And yet, we still are struggling to come up with treatments. In our lab, we're actually still trying to come up with treatments for HIV. And so in some of the ways that we're doing that is through a traditional method in our field known as the virtual screen, which is this top part. So we'll take all of these molecules that we think look like pretty good drugs, and then we'll try and shove them into our target and assess how well they fit, how well they interact, and do we think that they would make a good drug. And so we do this for millions and millions of molecules. And so we've found a lot of great drugs this way, a lot of cancer therapeutics this way, but it's a lot of unnecessary sampling that way. There was a big step in our field recently this month where someone had come through and actually screened 100 million compounds, and that was unheard of. But not a lot of people have access to these resources or this time. So an algorithm that was recently developed in our lab, which is called Novo Design, or Novo Design, is we take all of these millions of compounds that we would have screened, and we'll chop them all up into basically molecular Lego pieces, and then we're going to reassemble them in the target so that we're only sampling relevant chemical space, while maintaining some strict rules so that we're not defying the laws of nature, so that we can optimize and build drugs that are perfect for that target. And instead of doing a virtual screen, which for us would take 2 weeks, we can do this in a matter of hours. And so we'll have an ensemble of molecules that all have great properties towards these targets. So if you'd like to learn more about how we utilize this de novo doc algorithm to leverage the information that we found from heartburn medication to actually build potential drug candidates for HIV-1 treatment, feel free to come stop by my poster. I'm way in the back somewhere. Awesome. Thanks so much.